Deep within modern genomes, researchers recently claimed to find the traces of an ancient catastrophe. A high-profile analysis proposed that roughly 900,000 years ago, the human population crashed to exactly 1,280 breeding individuals. Almost immediately, that claim was fiercely contested in print by multiple rival research groups. Yet all of these disputing teams were extracting their entirely contradictory conclusions from the exact same public data source. That shared data source is known as the site frequency spectrum. The site frequency spectrum is, at its core, a simple histogram. It tallies how many genetic variants appear exactly once across a sample of modern genomes, how many appear twice, 3 times, and so on. This creates a direct scientific paradox. We have multiple mutually exclusive historical realities being derived from a single, unchanging histogram. The root of this conflict lies in a structural vulnerability common to standard bioinformatics pipelines, a massive dimensional mismatch between the raw data and the final conclusion. A population curve represents thousands of generations of continuous, highly detailed history. The site frequency spectrum is a comparatively tiny mathematical object condensed into a few discrete blocks of data. To bridge that immense gap, researchers rely on fitting software, using algorithms to stretch the simple histogram into a complex demographic narrative. A new identifiability theorem systematically dismantles this approach. It proves exactly what site frequency spectrum data can mathematically say about population history and what it cannot. By discarding algorithmic curve fitting in favor of mathematically certified bounds, this new framework reveals that the 900,000-year crash is largely an artifact of modeling choices, rather than a reality fixed by the DNA. Resolving this dispute requires discarding best-guess algorithms and demanding mathematical proofs of what the genomic data actually constrains. Establishing these proofs starts with defining the absolute mathematical limit of the information contained in the raw data. The fundamental rule is rigid. A sample size of n genomes yields exactly n-1 discrete numbers about population history. To project those numbers backward, geneticists use coalescent theory to translate discrete data points into historical time. Within coalescent time, each n-1 number functions strictly as an exponentially weighted average of historical population sizes. They are wide, overlapping blurs of history. Because the data only supplies these blurred averages, it mathematically cannot detect sharp time windows or exact population numbers at specific, isolated moments in the past. A historical quantity is only identifiable from the data if it can be built entirely out of a combination of those specific blurs. Therefore, any detailed curve shape or sharp severe population drop extending beyond those blurred constraints comes entirely from the fitting algorithm's internal assumptions. It does not exist in the raw data. To expose the danger of relying on those algorithmic assumptions, the researchers developed a constructive mathematical proof. They set up a controlled test, deliberately building pairs of genuinely different historical population scenarios. Take 2 opposing realities. History A features a severe population crash. History B is a continuous, stable curve. We track what happens mathematically when both distinct histories are processed through the coalescent bridge. The resulting histograms are absolutely identical, matching to 50 decimal places. The researchers globally certified the mathematical positivity of these constructed histories, proving that in realistic examples, they agree to 1 part in 20 million. The implication for the bottleneck claim is definitive. Because radically different histories output identical spectra, the genomic data is mathematically blind to the difference between a near-extinction event and a smooth, stable population. This forces a methodological pivot. Researchers must discard black box curve fitting entirely and replace it with mathematically certified bounds. Instead of guessing a single line, the new machinery uses linear programming to compute the full range of historical values that are actually consistent with the raw data. The model requires researchers to declare a strict complexity budget on admissible histories. It then outputs absolute minimum and maximum brackets rather than a single estimated point. The mathematical rigor here is absolute. Every endpoint generated carries an arithmetic certificate, computed to 50 digits, completely independent of the solver that produced it. To verify the entire data model bridge, a second implementation was built from a different mathematical route, using exact rational arithmetic. The 2 systems agree at machine precision. Furthermore, 8 deliberate corruption controls are permanently active in the code, confirming that these verification checks are live and functioning. Robust demographic inference must evolve into certificate-first research. Rigorous, identified bounding sets must replace the illusion of precision found in best-fit curves. With the machinery verified, the researchers applied it directly to the exact data driving the 900,000-year bottleneck dispute. The audit operated under a strict, pre-registered discipline. Every analysis choice, model grid, and cryptographic hash was frozen, published, and timestamped before the code ever touched the real data. Stage 1 of the audit analyzed the claimant's own Yoruba dataset, operating under standard pipeline rules that treat all genetic variants as independent observations. Stage 1 returned an unequivocal rejection. No clean single population history of any shape reproduces the raw dataset. This mismatch was extreme. The model could only find a fit by assuming the data carried far less information than actually reported. This tension localizes specifically in the high-frequency variant classes, revealing a signature of misassigned ancestral states, a common error in genomic processing that creates a false signal of a crash. This forced a pivot to Stage 2. The audit openly conceded these error channels, reducing the information budget to reflect the genome's actual recombination structure, instead of treating variants independently. Under Stage 2 settings, the final certified verdict on the Yoruba data is clear. The ancient population window is firmly bounded between 16 and 93% of the recent baseline. The ancient population was demonstrably depressed, but it sat certifiably far above the catastrophic 5 to 10% crash claimed by the original study. When the same audit was run on the provided European and East Asian genomic samples, the bounds certified no population reduction at all. We can now directly contrast the original headline claim against the mathematically certified bounds. The gap in evidence is immense. Successfully discriminating a severe crash from a smooth alternative would require 10 orders of magnitude more information than these spectra actually contain. The severity of the 900,000-year-old event was decided entirely by algorithmic modeling choices, not by the genomic data itself. Mathematical proofs now define the boundary of what can be known from the genome. By replacing uncertified curves with rigorous identified sets, researchers can finally separate genuine biological signals from the artifacts of their own software.